Hello friends, this video on molecular basis of inheritance part 28 is brought to you by examfear.com. No more fear from exam. So now the question is, who reads and deciphers the genetic code? Because now we got to know that there is a concept of genetic code based on which we will be able to tell the sequence of amino acids. And once the sequence of amino acids are known, then the amino acids can be joined together by peptide bond to form the polypeptides, which will form the proteins. Now the question is, who will do this job? So somebody actually needs to read the genetic code and then decipher the sequence of amino acid. So let us suppose this is the genetic code. So somebody has to read this genetic code and then give back the result. That is give the result of the sequence of amino acids. So who is going to do this? So this job is done by the tRNA, that is the transfer RNA, and that is why we introduced you to transfer RNA. It is called transfer RNA because it does the job of carrying the amino acids. So it is going to carry all the amino acids and it is also capable of reading the genetic code. So by reading the genetic code, it will get to know that which amino acid it should be, and then it carries that specific amino acid and helps in the process of protein synthesis. So that is the purpose of tRNA. So let us quickly try to understand the structure and behavior of tRNA. So as you can see, the picture shows how tRNA looks like. So it is transfer RNA. These are nothing but small RNA molecules with 75 to 90 nucleotides. So it is like any other RNA which has a single strand. So this tRNA molecule also is just like any other RNA molecule, which is a single strand of nucleotides, just that it is comparatively shorter than the normal RNA strands. It always has less than 100 nucleotides. So you can imagine it somewhat like this: a single strand of nucleotides which is being curved into to form a shape like this. So this was the traditional shape. This was the shape which was considered traditionally for the tRNA molecule, that is transfer RNA. So what is its purpose? It acts as carriers of amino acids. So it helps to carry the amino acid and then connects the amino acid to the correct codon on the mRNA. So let us see what exactly it does. So it plays a very important role during protein synthesis. And how does it do that? Let us have a look at that. Now let us suppose that this represents the sequence on the mRNA, and mRNA is the one which is being produced as a result of transcription from DNA, and the sequence of mRNA is somewhat like this. So this is just an example. Okay, so this is how the sequence is. So now based on this sequence, you need to find an amino acid. So who is going to do that? That is being done by the tRNA. So what will the tRNA do? tRNA will read this sequence. So tRNA will read it as AGU, and corresponding to AGU, whichever is the corresponding amino acid, it will try to bring that amino acid and then connect it to this. So let us suppose if AGU is for whichever amino acid, it will connect it to that. Again, U. Let us suppose it is for phenylalanine. So it will connect it to the phenylalanine. Again, CUU. It will see whichever amino acid it is. So it will connect it to that amino acid. So this purpose of connecting or linking the amino acid with the codon on the mRNA, this job is being done by transfer RNA. So tRNA acts as a mediator or a middle line between mRNA sequences and the sequence of amino acids which will later form the proteins. So it can connect the mRNA sequence with amino acid, and that is the main purpose of tRNA. So now let us talk about the structure of tRNA, because the structure is extremely significant, and it is because of this unique structure tRNA is able to perform this role. So earlier it was assumed that tRNA has a clover leaf structure. So this is how a clover leaf structure looks like. So as I said, you can imagine this structure as something like a straight strand being curved to form this shape. So, but As per the X-ray crystallography later, the actual structure was found to be an inverted L shape. So this is the inverted L shape. So this is how the structure looked like when it was observed when it was found out by X-ray crystallography. But this is the traditional clover leaf structure. So if you look at the structure of the tRNA closely, there are two important parts of tRNA. So let us discuss those two important parts. So one is the anticodon loop. So where do we have the anticodon loop? This portion is the anticodon loop. In fact, here and in this structure, this portion which you see here is the anticodon loop. So what is there in this loop? It contains the bases complementary to the sequence on mRNA. As I said, that the purpose of tRNA is to act as a link between the mRNA and the proteins. So what will this do is, here these three which you see here, these three will be complementary to the sequence which is present on the mRNA. So let us suppose if this is the mRNA and it has sequences like this. So let us suppose these are the sequences on the mRNA. So what is going to happen is, when a tRNA comes here, so the anticodon loop of tRNA will be this side. So the anticodon loop will have complementary bases when compared to the bases on mRNA. So as a result, they both will tend to come near each other, whereas the other end of the tRNA will consist of the corresponding amino acid, and this is how it will connect the right amino acid to the right codon, and this is how the connection established. So the anticodon loop will actually connect to the mRNA because of the complementary base pairing. So no tRNA exists for stop codons, but if there is a stop codon which is encountered, so for that there is no tRNA that exists. Initiator tRNA exists for initiation. So for every time you have that AUG, that is for methionine, so that acts as the initiator. That for that there will be an initiator tRNA. On the other end we have the amino acid acceptor end. So here you see this is the amino acid acceptor end. Or if you try to look at the inverted L structure, there also you see the same thing at the side. So here you see it connects to the amino acid. So these are the two ends which are extremely important, because through this end it connects to mRNA and on the other end it connects to amino acid. So it acts as a link between amino acid and the sequence on mRNA. So here tRNA binds to specific amino acid. Now each tRNA binds to a specific amino acid. So one particular tRNA will have one amino acid at its acceptor end. It cannot have more than one amino acid. So this is how the structure of, now please remember that at least one tRNA has to exist for one amino acid. There can be multiple tRNAs existing for one amino acid, but at least one has to be there. Now tRNA is termed as an adapter molecule. Why is it so? This is because of this special property of tRNA, that it can, on one hand it can connect to an amino acid, and on the other side it can also connect to the mRNA. So due to this, it is often termed as the adapter molecule. So it is termed as adapter molecule because it can read the mRNA sequence on one end and it can write the amino acid sequence on the other end. So it does a two-in-one job. So this side is the amino acid and this side is reading the mRNA. So that is why it is called adapter molecule. Now we will see that tRNA plays one of the most crucial roles during the process of translation, because I hope you are able to understand now. You will be able to relate it yourself that how tRNA plays an important role in translation, because translation is all about synthesizing proteins. Now to synthesize proteins you need amino acid. The tRNA will help you to sequence the amino acids. You will know which amino acid should come after which amino acid depending upon the mRNA sequence. Now after that, to create proteins, all that is needed is a peptide bond formation between all the amino acids, and that's all, you get proteins. tRNA is called transfer RNA. That is the full form of tRNA. Why? Because it transfers individual amino acids to the growing polypeptide chain, which later forms proteins. As I said, that tRNA will continuously keep on adding one amino acid. So it is actually trying to transfer the amino acid to the polypeptide chain. That is the chain consisting of many peptide bonds. So that chain is going to form proteins. So that is why it is called transfer RNA. So this is how the picture would be. See, these are the blue colored structures which you see, these are the tRNAs, and here the red structure is mRNA. So in mRNA you have a particular sequence. Now as the tRNA comes, it reads this sequence, the complementary sequence is added here, and the corresponding protein is present here. So it passes on the protein to this polypeptide chain. Again this moves on, the next one comes, and again for the corresponding sequence it will pass on the proteins to this chain. Again the next one will come, and as a result the transfer RNA will keep on transferring one amino acid to the polypeptide chain, and that is how the polypeptide chain will keep on growing and proteins will be formed. So let us look at some of the examples of tRNA as an adapter molecule. Let us suppose that the sequence on mRNA, let us suppose this is mRNA, and the sequence of bases on mRNA is something like this: AUG, UAC, UUU. So this is the sequence of bases on mRNA, and let us suppose this is a tRNA. So this tRNA, what will be the sequence of bases on the anticodon end? The sequence of bases on the anticodon end will be UAC. And what should be the amino acid on this end? So the amino acid for AUG is methionine. So this should be methionine amino acid which is connected to this end. Similarly for the amino acid UAC, so this will pass on the methionine then to the polypeptide chain, and it will move on. Then the next amino acid, the transfer RNA will come into picture. So here the mRNA is UAC. So what would be the corresponding bases on the anticodon loop? That would be AUG. And for UAC, which is the corresponding amino acid, the corresponding amino acid is tyrosine. So on the amino acid acceptor end, tyrosine will be attached. Similarly, if the next one is UUU, what would be the anticodon loop sequence? It would be AAA, and AAA is for, now this UUU is for phenylalanine, so this is going to be phenylalanine. Now let us suppose that after this, even if this continues further, let us suppose that after this the sequence is UGA. So this UGA is for a stop codon. So for stop codon you do not have tRNA. So there will be no tRNAs, and as a result the synthesis of the polypeptide chain will stop here. So basically in the polypeptide chain, first methionine will get added, then with peptide bond tyrosine will get added, then again with another peptide bond phenylalanine get added, and after that it will stop. So no more will get synthesized. So this is how the polypeptide chain formation will take place. So I think we have partially discussed the process of translation as well. So you have got a fair idea about how the process of translation is going to take place. But however, we will discuss translation in little more detail. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.